She received her degree at Johns Hopkins University in biophysics, and I have known her since she was a graduate student there. Then she became an assistant professor at UC Irvine, and after 16 years in UC Irvine and rising up the ranks to become full professor, she then moved to Caltech in 1996 and became the chair of the faculty at Caltech. And she was the first woman to hold that position, and she held that position for 2 years. So Dr. Bronner has done seminal work in the field of developmental biology, and her lab has had a long-standing interest in neural crest, lineage specification, migration, and differentiation. Her early work pioneered the use of single-cell lineage labeling approaches for analyzing the developmental potential of neural crest cells in vivo, and to show that the pre-migratory and migrating neural crest cells are multipotent. She then went on to define several of the signals underlying their induction, such as Wnt and segmental migration, for example, neurofilan. And more recently, her lab has systematically exploited this knowledge together with systems level transcriptional profiling, phylogenomic analysis, and in vivo functional perturbation experiments to reveal direct connections in the gene regulatory networks responsible for neural crest formation and evolutionary origin. Her work has led to the understanding of a number of birth defects related to defects in neural crest development, such as autonomic neuropathies and craniofacial defects. And she has published more than 300 papers in top journals. She's the editor-in-chief of Developmental Biology and on the editorial board of many journals. And for her outstanding achievements, she has won numerous awards, among them that this year she was elected member of the National Academy of Science. In 2013, she won the Cochrane Medal from the Society for Developmental Biology, and she has received the American Society for Cell Biology Women's Award in Cell Biology as a senior awardee in 2012. And she was also elected Fellow of the American Academy of Arts and Sciences in 2009. So the title of her talk today is Gene Regulatory Interactions Mediating Formation of Neural crest stem cells. So I'm sure you will all enjoy her talk today, and without any further ado, I present to you Dr. Marianne Bronner.
Thank you very much, Peng, for that lovely introduction, and it's a great pleasure to be at the NIH. I always love coming here to Washington to see rain, which we don't have in California. Please ship us some. So as Peng mentioned, I'm going to tell you about our work on the neural crest, which I've been working on for an embarrassingly long period of time. And the neural crest has always fascinated me as an interesting cell type that gives rise to a variety of different cell types in the embryo and in the adult. And one of the reasons that I like to study them is because I love embryos. They're very beautiful. So if you see this, can you turn the lights down just a bit, please? So this is an image of an embryo viewed from above, where the nervous system is here. This is the brain, and going down to the spinal cord. Neural crest cells arise from the developing nervous system, but then they do something remarkable. They migrate out, and here you see them migrating in this beautiful stream. So we like to study how these cells form, and we can also follow their migration in living embryos, as you see here. And we can learn a tremendous amount by studying how they leave the neural tube and how they migrate to distant locations. Once they reach their final sites of localization, neural crest cells differentiate into a vast number of derivatives. They form all of the pigmentation of our skin, much of the craniofacial skeleton, portions of cranial ganglia, and other elements of the peripheral nervous system, including enteric nervous system, sensory nervous system, and many other derivatives. So they give rise to a wide variety of cell types. So one of the reasons we're interested in them is because we want to understand how a single stem cell population can give rise to such a diversity of derivatives. Of course, we're here at the NIH, and so one of the reasons that at least I tell my granting agencies that I want to understand neural crest development is because defects in neural crest development are actually one of the most common birth defects. So about a third of all congenital defects involve the neural crest in one way or the other. In addition, the neural crest cells, when they go bad in the adult, give rise to many types of cancers. And because they're such an invasive cell type in the embryo, we think that some of the programs that they go through normally during development may have a role in causing neural crest-derived cancers like melanoma and neuroblastoma. Most recently, we've been very interested in the possible regenerative potential of neural crest cells, and we'd like to know if we can take these early populations of cells and, by understanding their regulatory programs, cause them to differentiate into particular types of derivatives or give rise to self-renewing progenitors for the purposes of regenerative medicine. So these are many good reasons to study the neural crest, but really the reason I'm most interested in is because they're such an interesting and beautiful cell type. So what I'd like to do today is start at the very beginning of neural crest development and tell you a little bit about how the neural crest cells form and how we're trying to study the molecular aspects of their formation. So this is a schematic diagram showing the early embryo at the time that the embryo has 3 germ layers. So this is the top germ layer, or ectoderm, and the ectoderm will give rise to the nervous system, the skin, and the neural crest. So in the midline of the embryo is a flat tissue called the neural plate, which will fold to form the central nervous system. So this tissue invaginates, transforming from a flat sheet into a cylindrical neural tube. And this neural tube will then give rise to the brain in the head and to the spinal cord in the posterior portions of the embryo. Lateral to this neural plate is the non-neural ectoderm, shown here in white, and that will form the epidermis. And between these two tissues, between the presumptive epidermis and the presumptive nervous system, lies this region which I'll be referring to as the neural plate border, and these contain the presumptive neural crest cells. So during the process of neural tube closure, this green zone here, this neural plate border, forms the leading edges of the closing neural tube. After the neural tube closes, the neural crest precursors are initially contained within the central nervous system, and there are actually neuroepithelial cells. However, they do something quite remarkable. They lose their epithelial connections, and they undergo something called an epithelial to mesenchymal transition to migrate from the neural tube and become migratory neural crest cells that move to distant locations in the embryo. And so here is just a section through the embryo where the cells start life in the dorsal neural tube and then migrate out along distinct pathways to many sites in the periphery. There they differentiate into many different cell types. So neural crest precursors give rise to much of the cartilage and bone of the face, many neurons and glia of the peripheral ganglia, and all of the melanocytes that are in our skin. However, the neural crest population along the body axis is not homogeneous. Cranial neural crest cells, in fact, are the only ones that give rise to cartilage and bone of the face, whereas those from the trunk region give rise to many derivatives, including neurons and glia of sensory ganglia and melanocytes, but they fail to give rise to cartilage and bone, even if you take this piece of neural tube and transplant it into the head region. So there are interesting differences along the body axis with respect to the types of derivatives that neural crest cells form. And this is something that has intrigued us for many years and will be part of what I'm going to tell you about today. So we've been studying the neural crest for many years now, and about a decade ago, we tried to understand this from a more, what we would today call a systems biology approach, and that is to try to understand the molecular building blocks that are functioning at different stages. And so we call this assembly of a gene regulatory network, and it started just by looking at individual genes that are expressed in this neural plate border region, and we found that in many different vertebrates, the neural plate border genes like Pax7, Msx, and Msx were co-expressed in this neural plate border. We also knew that signaling molecules were important for establishment of this border region, and that signals like Wnt and BMPs established this region and perhaps turned on these transcription factors that were important for early stages of neural crest formation. We then speculated that these signals and transcription factors somehow worked to turn on genes that became expressed just as the neural tube was beginning to close, and we call those neural crest specifier genes. And those include genes like snail, FOXD3, SOX10, and many others that are expressed very early as the nervous system is forming and are the first markers that distinguish this population from other cell types in the central nervous system. And then we speculated that these genes, in turn, control other factors that are important for causing these cells to undergo an epithelial mesenchymal transition, to start to migrate, and then ultimately differentiate into many different derivatives. So I'm going to use this platform as a scaffold to tell you how we're attempting to study the neural crest system. And I'll start just by describing some old experiments in which we looked at one of these neural plate border genes, Pax7, to try to understand what it might be doing in the process of neural crest formation. So this work was done by a former postdoc of mine, Martin Garcia Castro, and what he was really looking for was just a marker that would describe this early neural plate border in a nice and early fashion. So what he found was Pax7, shown here in red, was expressed very early on in this neural plate border region and seemed to beautifully define and outline where the presumptive neural crest cells would come from. So in order to analyze what this might be doing in the embryo, we wanted to understand the function of Pax7, and we'd speculated that Pax7 was somehow upstream of these neural crest specifier genes. So we looked at its function by doing a knockout experiment. And I should mention that the first experiments I'm going to tell you about in my talk are done in bird embryos. And I know a lot of people here work on mice, and mice are beautiful and wonderful animals. But it turns out for neural crest development, they're much more difficult to work on. And a lot of the data that we've gotten to help us understand early neural crest development actually has come from studies of other vertebrates like chick, zebrafish, and frog. So we wanted to knock down Pax7, and so in the chick, we actually have a nice trick that we can use to knock it down on one side of the embryo. So this is a gastrula stage embryo. This is the primitive streak. And we know from fate mapping that the neural crest forming region is about here. So here we can put an inhibitory molecule, and in this case we're using an antisense morpholino. And we know by fate map that this will be inherited by the neural crest cells on one side of the embryo. And this is nice because we can use the other side as an internal control. What we found was when we knocked down Pax7 is that we in turn lost expression of neural crest specifier genes, including SNAIL2, which is shown here, SOX9, SOX10, and FOXD3. So going back to our diagram, what this showed us was that Pax7 was in fact upstream of all these other regulators. And that was a way for us to validate this presumptive network. So that's nice because it tells us there's order in our network and that these neural plate border genes are upstream of our neural crest specifier genes. But what it didn't tell us was if Pax7 was regulating these downstream factors in a direct fashion or in an indirect fashion. So the next step was to try to look at direct interactions in our network. And the way we did that is by taking a view from the genome up. And I'll just show you one example of how we can do this with a neural crest specifier gene called FOXD3. So we chose FOXD3 because it's one of the earliest genes to be expressed as the neural tube is beginning to close. And about the time that we started these experiments, the chick genome was sequenced, and it turned out that it was an optimal distance away from mammalian genomes to allow easy identification of conserved regions. And as you might expect, the coding region of a gene like FOXD3 would be highly conserved. But what we found is that in addition to the coding region, when we lined up many different genomes from human to mouse, to rat, to opossum, and chick, in the intergenic regions, there were also lots of peaks of conservation, which are shown here. And if they were conserved in the intergenic region, we suspected that, in fact, those might represent enhancer regions, conserved regulatory regions. So then we went back to our chick embryo, and the nice thing about using the embryo is we can easily test these regions to see if they have enhancing activity in the neural crest. So basically what we do is we take these conserved regions, which we call putative enhancers, we hook them up to a basal promoter that encodes either GFP or RFP, and then we go back to our embryo, and just like we did with the morpholinos, we basically use electroporation to zap these constructs into the entire embryo, and then we look 1 day later, and if we have a positive, it looks like this. So this is an embryo viewed from above. This is the forebrain, here's the midbrain and the hindbrain, and here is the cranial region of the neural crest, where you see these beautiful migrating neural crest cells expressing GFP. And what that means is that this putative enhancer actually was mediating reporter expression in a pattern that overlapped with the neural crest. So this is a way that we can identify putative enhancers. And I just wanted to point out that even for those of you who do work on mammalian embryos, this is a fantastic assay for not just chick enhancers, but also we can take just about any species. We can take human enhancers, mouse enhancers, and the conservation is such that they work perfectly well in the bird embryo. So it's a very fast, easy, and very inexpensive assay in comparison to transient transgenesis in the mouse. Right, so the reason we did this experiment was to look at FOXD3, and so FOXD3 coding region is here. This is the next adjacent gene. So we tested all these blocks of putative regulatory regions, and what we found is that 2 regions, which we called NC1 and NC2, actually had enhancing activity. So that was very exciting, and it meant that our assay worked. But interestingly, the enhancing activity was not the same for these 2 different enhancers. The first enhancer, NC1, recapitulated endogenous FOXD3 expression in the cranial region of the neural crest. So this shows the in situ pattern of FOXD3 in the cranial crest, and you see that it exactly matches the pattern that we got with electroporation of the enhancer. But the other region, NC2, actually mediated expression not in the head, but rather in the trunk region. So this was very interesting and also very exciting to us because it suggested that encoded in the genome was information that was cranial specific or trunk specific regarding the expression of this transcription factor, FOXD3, was actually expressed along the entire axis. So the next thing we did once we had these enhancers was found a minimal enhancer, and then we wanted to look to see what might be mediating expression of that enhancer. So I'm going to cut through lots of hard work and just show you what the minimal enhancer looked like, because then we can just look at the sequence by bioinformatics and look for putative enhancer binding sites. And what you see here is all the different binding sites that we found using programs like Jasper and others. And then what we do is we basically test each of these sites by mutating them individually. And many of these sites don't have any effect at all, but what we found is that when we mutated a Pax site here, an Etst site here, or an MSX site, individually, mutation of any one of these sites caused us to lose enhancer activity, suggesting that all 3 of these inputs were necessary. And so that's shown here. So basically, this is the control region where we have enhancing activity, and if we mutate any one of these sites, we basically lose that activity. So then that is very nice because it suggests that binding to these regions may be Pax7, which is what I had predicted might be an input into neural crest specifier genes. But many things can bind to a transcription factor binding site, so this does not prove that Pax7 is the binding site. So then the technique that we next use to really identify what the direct input is going back to our electroporation experiments. Here we have our embryo, and we basically take our wild-type enhancer and put a morpholino, co-electroporate it with a morpholino on one side of the embryo, and on the other side, we take our enhancer with a control morpholino. And so the embryo then looks like this. If, in fact, our targeted morpholino knocks out the right thing, we should see a pattern where we have control activity here and diminished activity on the side that inherited the morpholino. So what we found then when we did this kind of analysis with NC1, the cranial enhancer, is that Pax7, MSX, and Etst1 were all critical binding sites and critical inputs into this enhancer. We did exactly the same type of analysis for the trunk enhancer, and interestingly, we found some common inputs from Pax7 and MSX. However, Etst1 had no effect on the expression of this trunk enhancer, and in contrast, another neural plate border gene, ZIC1, seemed to be important. So just to summarize this kind of enhancer analysis, which we've now done for many different genes, what we found is along the body axis, expression of the neural crest specifier gene FOXD3 was controlled by 2 different enhancers, NC1 and NC2, that had some common inputs, these neural plate border genes, Pax7 and Msx. However, the cranial enhancer had an additional input that was required from Etst1, another transcription factor, whereas the trunk enhancer didn't require Etst1, but instead required Zik1. And this actually made sense to us when we looked at the distribution pattern of these different transcription factors. What we found is that Pax7 and Msx were expressed all along the body axis, whereas Etst1 turns out to be a cranial-specific transcription factor, whereas Zik1 is specific to the trunk. So that suggested that this regulation had both common and different inputs. And then we went on to verify this using chip analysis. And in fact, all the things I talked about are direct inputs into these different enhancers. So the reason I show you this is because this allows us to really elaborate our gene regulatory network that's working in both the head and the trunk region. And what we've looked at is inputs into FOXD3. And this in-depth type of analysis has allowed us to understand that these inputs are actually direct in our network. The other nice thing is that using this kind of analysis, we actually found transcription factors that we didn't really know were part of our network. For example, Etst1 had not been previously put in as a member of our network, and this showed that Etst1 was not only an input into FOXD3, but was also a cranial-specific factor. So that's a nice way to demonstrate direct interactions and to identify new things. So this is nice, and we've done a lot of analysis of this sort with many different enhancers. And so this has provided us a way to really construct this network in a much more detailed form. But what I'm really excited about is none of this, but rather the fact that now this gives us a great tool to further study the neural crest. And one of the challenges of studying the neural crest is that neural crest cells initially start out in the dorsal neural tube, but then they move all over the embryo. And this is a problem if you want to understand what factors are present in this isolated population. By using these enhancers as a tool, however, we can now sort discrete populations of neural crest cells, either at pre-migratory, migrating stages, or even very latent differentiation. And this gives us a handle on further exploring the regulatory landscape that's present in these cells. So basically what we do now is we can use these enhancers and then sort the cells at many different stages, either early in the neural plate border, when neural crest cells are pre-migratory, or when they're actually migrating through the embryo. So this is work that was done by Marco Simões-Costa, a wonderful postdoc in my lab. And what he's been doing is transcriptome analysis of all these different populations. And basically the technique is to do exactly what I showed you before, electroporate the enhancer into early embryos. We then FACS sort them and perform RNA-seq. And this is very standard, and I'm sure many of you are doing similar types of things. But I was really excited about it because when every time a new technique came about to somehow be able to isolate the transcriptome of cells, we tried it. For those of you who are older in the audience, we tried differential display, which is probably like 20 years old. We've made macroarray libraries, and we've done lots of things. And we found lots of interesting genes, but very few transcription factors came up in these analyses. And so what I was really heartened by was that by doing RNA-seq, we found 1,300 genes that were upregulated in the cranial neural crest compared to the whole embryo. And of these, 50 were novel transcription factors that we had not previously put into our network. So that was good news on 2 counts. First of all, we got a lot of transcription factors. And secondly, we didn't have so many that we couldn't possibly analyze them all. 50 is a reasonable number to study. Right, so then we did this nice transcriptome analysis. We wanted to get the paper published, hopefully in a good journal. So we did all the kind of things to make it look pretty. And one of the things we did was this biological pathway analysis, which isn't really that interesting. But I wanted to point out one thing, and that is that we found lots of genes that were upregulated in Wnt signaling. And we'd known for many years that Wnt signaling was important in neural crest formation. So I was very interested to see this large upregulation in factors that were involved in the Wnt pathway. And this took me back to some experiments that we did many years ago to look to see what kind of signaling molecules were important for neural crest formation. So these are days when we were doing mostly cut and paste biology. And what we found, if you look at the embryo, here's the neural tube as it's beginning to close. And neural crest cells normally form here. And that point is at the juxtaposition of the neural and the non-neural ectoderm. So it didn't take a rocket scientist to think that perhaps there was some kind of interaction going on between these two tissues, the presumptive epidermis and the presumptive neural tube. So we took a region of the neural tube that's far away from the normal neural crest forming region, and we put it together with the region of ectoderm that's also far away from where neural crest cells normally form. And when we do that and put them either in culture or let them stay in the embryo, what we find is that juxtaposing these two tissues, neural and non-neural, gives us a new cell type, the neural crest. So this is like a classic inductive interaction, suggesting that there's signaling going on between these two tissues. We then did a candidate search to look to see what might be involved, and what we found is that Wnt was very nicely expressed in the ectoderm at just the right place and time to be responsible for this signaling. We then blocked Wnt signaling here, and we found that we no longer got neural crest formation. And we further took a piece of the neural tube from here, which normally would never form neural crest, and if we add to it Wnt, then we got neural crest cells. So that led us to the, we now realize, sort of naive conclusion that Wnt signaling was all you really needed to get neural crest. So anyway, the point is that we knew Wnt was somehow important in neural crest formation. So going back to our regulatory network, that accounts for why we thought Wnt signaling was important in this place. And what I was really excited about when I saw the RNA-seq data is that one of the genes that was upregulated in our screen was actually one that was thought to be a Wnt-effector gene. So we wanted to analyze whether this gene, which I'll show you in the next slide, it's called AXID1, might be somehow involved in linking the signaling module to the neural crest specifier module. And so AXID1 is expressed early in the neural crest, as shown here. And from the literature, we found that Phil Soriano's lab had knocked it out in mice, and it had a neural crest defect in the palate. So that made us wonder if, in fact, it might be important for expression of some of these neural crest specifier genes. And in fact, we found that AXID was beautifully expressed in earlier than FOXD3, but in a pattern that nicely overlapped with it. And when we did double in situs, in fact, what we found was that AXID, the FOXD3, came up within the AXID domain, but shortly thereafter. So what we then wanted to do was test to see whether AXID was important for FOXD3 expression. And so the way we did that is exactly the same experiment that I've shown you many times before now, and that is to electroporate something, in this case a morpholino against AXID1, into one side of the embryo, whereas the other side served as an internal control. And what we found is that when we lost AXID expression, we lose FOXD3. And if we do the opposite experiment, where we ectopically or express it early in neural folds, we get premature FOXD3 expression. Interestingly, shown here, we not only had an effect on endogenous FOXD3, but this FOXD3 enhancer that I showed you previously was also expression mediated by that enhancer was eliminated when we knocked down AXID. So I showed you this before, where we looked at the sequence of this enhancer for putative binding sites. We really didn't know that AXID existed when we first did this, but now we went back, and what we found was that, in fact, there was an AXID1 binding site in between the Etst and the Msx binding sites. And when we mutate that site, in the same way we did previously with other experiments, we actually lose FOXD3 enhancer activity. And then we went on to do chip analysis and found that, like Pax7 and Msx, AXID was also a direct input into this FOXD3 enhancer. So that was intriguing to us, and it made us think that AXID may really be a direct effector gene of Wnt signaling. So to verify that, we looked more closely, and when we looked through sections through the embryo, so here's the neural tube, and this is the dorsal neural tube where neural crest cells arise. When we look at Wnt1 expression, it's just where we expect it, and AXID is expressed exactly within that Wnt domain. We then knock down Wnt, and we find that we lose AXID, And the same thing happens when we knock down beta-catenin, suggesting that AXID is somehow mediating canonical Wnt signaling. So what this showed then is that AXID was downstream of Wnt. But if in fact AXID is the Wnt-effector that's responsible for turning on neural crest formation, then we would expect it to be able to rescue. So this is the critical experiment, because we know that Wnt is important for neural crest formation. And in fact, when we knock down Wnt, we lose neural crest formation, as shown here. But what we found is that when we knocked down Wnt but added back AXID, that was sufficient to rescue the phenotype. So that really puts AXID as the downstream effector that mediates expression of genes like FOXD3. And we've actually done this with a number of other neural crest specifier genes as well, and it works in a similar fashion. Right, so that suggests that AXID is really important for turning on this neural crest specification module. So that was really nice, and it sort of validates the fact that these RNA-seq screens can give you nice new genes that you can put into your network. But there was also one other puzzle that we were hoping this would solve. And when we formulated this gene regulatory network, we knew that there were these transcription factors at the neural plate border, and we knew that there were signaling molecules, and we knew both of these were somehow important. But what we didn't know is how these modules interacted with one another. So because AXID was important for turning on the same genes that Pax7 and Msx were responsible for, we wondered whether there was some link and that AXID may be integrating Wnt signaling and these neural plate transcription factors. So we wanted to test that to see if, in fact, maybe they might be interacting with one another. And to do that, we used a technique called proximity ligation, which allows us to look in vivo at interactions between different pairs of transcription factors and other molecules. So this just shows the results, where here we're looking at the dorsal neural tubes in all of these pictures. And in this case, we looked at interactions between AXID1 and Pax7. And when you see an interaction, you see a red dot. And here we've enhanced the dots so you can see them more easily. And what we found is that Pax7 and AXID did, in fact, interact, as did AXID and MSX. As negative controls, we used other genes that were expressed in the neural tube, like Etst1 and snail, and what we found is that they did not interact above what background levels that you would see in other parts of the neural tube where these transcription factors are not present. So this suggested that in vivo, AXID was in fact interacting with Pax7 and MSX. So our model then is that AXID is really a missing link that allows this neural plate border module to integrate with the signaling module and turn on neural crest specifier genes. And our results show that these then go on to the enhancer region of genes like FOXD3 and mediate its expression, and that each of these components is required. And we actually found that if we eliminate AXID, we no longer have binding of Pax7 or MSX to this enhancer region. So what I really like about this story is that we really took sort of an approach where we looked for genes that were expressed in the neural crest by transcriptome analysis, but by picking particular candidate genes like AXID and looking in depth and performing an in-depth analysis, we were able to solve a long-standing puzzle in the neural crest field, and that is how you mediate the interactions between signals like Wnt and transcription factors at the neural plate border like Pax7 and MSX. And what the data show is that the canonical Wnt signaling components interact with or mediate AXID expression, and this in turn turns on FOXD3 expression to turn on neural crest formation. So I was very excited about this because I think it gave us a great inroad into our regulatory network and shows how we can take the information gained from transcriptome analysis and really get a better understanding of the system. So emboldened by this, we went back and we wanted to really try to understand what was, other components of the neural crest network. And everything I've shown you so far has been looking at the cranial neural crest. And we've really focused on that basically because it's easier. But one of the things I'm very intrigued by is why are there differences in the potential of neural crest cells at different levels of the neural axis, such that cranial neural crest cells uniquely give rise to cartilage and bone of the face, whereas trunk neural crest cells ordinarily cannot do so. So if transcriptome analysis works so nicely for the cranial crest, we thought it would also be a very nice way to compare cranial versus trunk. So Marcos then went back and basically repeated this same type of transcriptome analysis, looking at genes that were expressed in the pre-migratory cranial versus the pre-migratory trunk neural crest. And we really focused on the cranial neural crest genes because we want to understand why they can make cartilage, whereas trunk cells cannot. And we were very happy to find, again, novel transcription factors in the pre-migratory neural crest. And in this case, we found 15 transcription factors that were uniquely expressed at this axial level. And this is really great because our experiments are largely done with morpholinos. Morpholinos are pretty expensive, so if we found even 50 would be hard to work with, but 15 was manageable within our budget. So what I'm going to show you then is how we're taking this kind of analysis and trying to use these new genes that we're finding and try to order them in our regulatory network. So we found a lot of beautifully expressed genes. This is brain3, DMBX1, LHX, and as I already showed you, AXID, all of which are cranial specific. So what we want to do then is try to use these and try to use both the timing of their expression and functional analysis to put them into a network. So I just showed you that with AXID that we found that AXID was a mediator of FOXD3 expression. And what we had previously shown because of our enhancer analysis that Etst1 was also an input into FOXD3, specifically in the cranial region. So then we have these novel factors. So the DMBX1 is expressed slightly after brain3, and it's expressed very nicely in the early neural folds. So then what we do is we just test what it does in the same way I've shown you by doing a knockout on one side and a control on the other. And what we found is that when we used Etst1 as a readout, we lost Etst1 expression when we knocked down this gene. So in terms of our network, that puts DMBX1 upstream of Etst1. So then we went on and we looked at another factor, MIB. And MIB was a very interesting gene because we found that when we knocked down MIB, we also lost Etst1. So that suggested that MIB was an input into Etst1. So we sent this paper off to review. It was just a short paper, sort of a leftover of one of my graduate students' projects. And we sent it off, and the reviewer said, well, you looked at a few genes, but what about other interesting genes like ZIC1? Why don't you look at that? So we begrudgingly did the experiment, as one has to do for reviewers. And what we found was really interesting. Although MIB1 seems to be an input into Etst1 that's positive, it actually inhibited ZIC1 expression. So the reason this was interesting is I told you in terms of inputs into FOXD3 that Etst1 was an input in the cranial region and ZIC1 was an input in the trunk. So early on, ZIC is actually expressed in the head, but when MIB comes on, it gets turned off. So we're very grateful to our reviewer, in case you happen to be out there, because by proposing this experiment, we actually have a much better understanding of how this network works. So then we went on to look at other genes, and one of the earliest genes that was expressed was brain3. And when we knocked out brain3, we found that we lost DMBX1. So this is both temporally and spatially, this seems to be the earliest gene that comes up in our cranial specific network. And we've also looked downstream at later genes that are expressed, and we found that ALX seems to be important, and ALX is downstream of Etst1, and this is important for cartilage formation. So I know I went through this very quickly, and I did that intentionally, but this is meant to just show you how by doing some relatively simple experiments and taking advantage of the expression patterns, you can begin to assemble what looks like a pretty decent regulatory network. Right, so why do I want to do this? Well, one of our goals in terms of regenerative medicine is to see if we can program cells like naive neural crest cells to make cell types like cartilage and other cranial specific cell types. So what we'd like to do is test whether by taking components of this network, we might be able to convert cells from naive cells or from iPS cells into particular lineages. So I'm just going to show you briefly how we're starting to try to do that. And we call this reprogramming because that's the sort of interesting word right now. But basically, here's our embryo. And I've shown you that we have cranial-specific enhancers like the FOXD3 enhancer. And this normally comes on in the head. but fails to come on in the trunk region. So as our first step in reprogramming, what we are trying to do is just see if we can take that network that I just showed you and misexpress it in the trunk region and perhaps turn on this cranial-specific program. So we've made attempts to do that, and the first thing we did was we tried to express this very early factor, brain 3, and see if it would turn on the cranial enhancer in the trunk. And the bad news was it didn't work at all. But when we took this construct and then simultaneously did a loss of function for ZIC1, what we found is that the cranial enhancer now was expressed ectopically in the trunk region. So these are preliminary experiments, and we're continuing them. These cells do not go on to make cartilage yet, so we know that we need to add other components. But we think this is sort of an inroad into trying to use our regulatory information to then reprogram the neural crest. Okay, so everything I've shown you so far has been using the bird embryo, which I think is very good for looking at early steps in neural crest formation. And so I've shown you sort of 3 different stories. One where we did in-depth cis-regulatory analysis to look to see how genes in our network were regulated. And I showed you how we found that FOXD3 in the cranial crest was regulated by a combination of Pax7 and Msx together with Etst1, whereas in the trunk it was regulated by Pax7 and Msx together with a different transcription factor, ZIC. Using these enhancers that were axial level specific, we can then do a lot of nice transcriptome analysis, which gives my lab basically everything we need to do for the next 100 years or so, if I last that long. And what we found by then taking some of that transcriptome data and looking in depth is we found a new player, AXID1, and that gave us an exciting result showing that Wnt signaling was actually mediated by AXID1 that then worked together with these neural plate border genes to turn on neural crest specification. And finally, I showed you how we'd like to use the information we've gleaned from the earlier experiments to do reprogramming. And basically, we're trying to assemble a gene regulatory network that tells us what's different between cranial and trunk neural crest cells. And going forward, we'd actually like to do this for other neural crest populations as well. So this is what we've done to look at early events in neural crest formation. One of the things I'd like to do going forward is go later in time and look at how the cells are migrating and how they're differentiating. And to understand cell migration and differentiation, the chick system is good in some ways, but it's also problematic in that it's hard to visualize cells as they're migrating. You can do it for short periods of time, but later on it's hard to see them. So for the next step in this analysis, we've actually transitioned to largely using the zebrafish model. And I just wanted to show you some rather quick experiments to show you what we're trying to do going forward. And these are experiments that were done by another postdoc, Ankur Saxena, in my lab. And he got very interested in migrating neural crest cells and their contribution to different structures, and found an unexpected result, and that is that neural crest cells not only form the derivatives that we knew about that I showed you before, like pigment cells and different types of neurons, but they also contributed to elements of the nose and to a specific population of olfactory neurons. So this is a zebrafish embryo. Here's the head of the embryo, and the neural tube is here. And these green cells are migrating neural crest cells, and they migrate and populate almost the entire head region, and they surround the nose at later stages. So this is a slightly older embryo, and here is the nose that's marked with these in red, with a neurogenin probe. And so what Ankur found, just by looking into the zebrafish, is that if you look into the nose, this is an embryo viewed face on. So here's the neural tube, and here is the olfactory epithelium on the left and the right side. There are ciliary neurons that are shown here in red. But he found that with time, these green cells that are neural crest derived began to come in from the nasal cavity and invade the olfactory epithelium and become another population of neurons, which were called microvillus neurons. So we're interested in studying how these cells move from the neural crest into the nose and then differentiate into this very specific and beautiful population of neurons. So this is shown here, and the reason that I show you this in zebrafish is because it's so much easier to visualize. So this is another zebrafish embryo at a later stage. This is the olfactory epithelium, and these green cells are these beautiful bottle-shaped olfactory microvillus neurons in the nose. So we were really interested in following many aspects of development of these cells. We wanted to look how they got into the olfactory epithelium, and then how they differentiated into neurons, and finally how they put axons that went to the olfactory bulb. But here we got stuck, because using conventional confocal, you get a really nice face-on view of the embryo, but we really wanted to look in 3D. And so when you look at the olfactory bulb, you see these nice neurons, but if you take your image and rotate it 90 degrees, it really looks awful, and you can't see very much. So we were stuck here, and one of the things that I was talking to a lot of students about this, and postdocs, about how interesting the technology development has been and how that's helped our science. So here we thought perhaps by using new technology, we could actually do better and see deeper. So at about this time, Encore heard a talk from Eric Betzig's lab at Genelia Farms, one of your neighbors, and Eric had developed the lattice light sheet microscope, and so we thought that this might be a good opportunity to try to see if this would help us in our imaging. And so basically, he went to Genelia to do lattice light sheet imaging and to see if this could actually work on a vertebrate, because previously it had only been applied to invertebrate embryos, to mostly Drosophila. And the advantage of this, the light sheet, or the lattice light sheet, is that it has very high resolution in 4 dimensions, and it has very low phototoxicity. So this is actually one of the early images that he got. So each of these frames shows exactly the same olfactory epithelium with these invaginating neural crest cells that form neurons. And you can see from 4 different angles, it looks almost identical. So it's really very, very beautiful. So this just shows a comparison between what we were seeing with traditional confocal microscopy and lattice light sheet, which is just beautiful. And so this is I think a clear example of where modern technology can be addressed to really classical embryological experiments. And although we're still working on this and trying to understand different processes, we think this will be a great way to really understand neurogenesis of this population of cells. So basically, we're applying this to cell migration from the nasal placode into the neural crest. We want to see how these things are guided, how the axons make their way to the olfactory bulb. And the other really nice thing about this kind of technology is that not only can you look broadly, but you can also look deep and look at cytoskeletal elements and how they are changing with time. So that's very exciting. So I'm just going to end by showing you a very gratuitous movie. This is the nose of a slightly older fish, and we're just looking with lattice light sheet through different layers of the nose. And I'm only showing this because it's really, really pretty. You can see the cilia beating, and in fact, we had to slow the movie down 4 times in order to follow the cilial beating. So I'm just going to end there and summarize. So what I've tried to tell you about today is our neural crest gene regulatory network. And basically, we've been working on this in a number of different species. I mostly showed you what we'd found in the chick embryo. And I showed you that by analyzing enhancer regions of genes like FOXD3, we can learn a lot about direct regulation of genes within our regulatory network. We can then use these enhancers as tools to do transcriptome analysis, which gives us lots of new material in order to try to analyze this network in ever deeper depth. But in order to understand what's going on, you can't just look broadly. It's great to look at a systems level, and we all sort of say systems biology is the thing of the future. But really, to understand what's going on in detail, you have to look in depth. And so I showed you one example of how, by picking a candidate gene, AXID, which has been proposed to be an effector of Wnt signaling, we actually solved a mystery in the field, and we found that AXID actually integrates Wnt signaling with neural plate border transcription factors to turn on the neural crest regulatory program. And finally, I ended by showing you sort of where we're going in the future, and that is to look at how neural crest cells migrate and then differentiate into different populations of cells. And we've seen in zebrafish, unexpectedly, that neural crest cells contribute to a population of neurons, kind of microvillus neurons in the nose. And we're trying to understand the role of these neurons and how they act on pathfind. Right, so just to end, I want to acknowledge the people who did the work. Most of the work I talked about today was done by a fantastic postdoc in my lab, Marcos Simões Costa. He analyzed the FOXD3 enhancer regions together with Sonia McCune and Tatiana Sauka-Spengler. He has done the ACSID experiments that I showed you and has really done a beautiful in-depth analysis of this particular transcription factor. He's also done all the transcriptome analysis I've shown you and has been assembling this neural crest gene regulatory network. At the end, I talked about work done by Ankur Saxena, who worked together with Eric Betzig and Bi-Chang Chen at Janelia Farms to use beautiful lattice light sheet microscopy to better understand how neural crest cells migrate and differentiate. And I'd like to end there, and I'm happy to take any questions. Thank you. Can't even see out there.
Hello.
Hello.
At the beginning of your lecture, you mentioned, I know that you were going more for the reprogramming of cells, but you did mention a melanoma, neuroblastoma, and glioblastoma, multiform. Right. First one, melanoma is very diverse. Actually, you're talking about nevus cells, and that not only will go to a melanoma, but can go to a million other things that are interstate. The second one, the neuroblastoma, is essentially a pediatric tumor, which has the property that it can spontaneously regress. And the glioblastoma multiform is the end of a high end of a range of astrocytomas in the brain. Now, I can't really find any common denominator between them. They're all just very strange. In my experience in pathology, they're very strange actors at the periphery of everything. So in terms of your transcriptome, have you found anything that you could apply actually to the tumors that would serve in two manners?
Well,
one, immunoperoxidase diagnosis, and second, treatment.
Right. That's exactly where we're going. I mean, I went to an A&R meeting last year for neuroblastoma, and that's when I got really excited about neuroblastoma. And I was struck by the fact that people were doing lots of transcriptome analysis, but they didn't really know what the normal profile of the cells should be. So one of the things we want to do is profile those sympathoadrenal precursors and see what type of neuroblastoma they may resemble. And my hypothesis is that the more neural crest-like they are, the more aggressive the tumor will be. But I have no data. But that's what I'd like to know. We certainly know, I mean, I didn't talk about this today, but genes that are always mutated in neuroblastoma include NMYC and CMYC. And we've been studying how MYC, I didn't have time to show all of our data, but MYC has an important function in the early neural crest that relates to defects in cell migration and could really be involved in metastatic behavior that would correlate at least to what happens in neuroblastoma. So I mean, we're just beginning these studies, but what my hope is I think that the tumors that are most aggressive are going to be those that most resemble the least differentiated neural crest cells. And so if that's the case, by understanding the normal transcriptional program, we may be able to find some therapeutic targets. But that's sort of years down the line.
But the diagnostic aspect would be most important in melanomas.
Right.
The other ones are not really a diagnostic challenge, just to mention that.
Well, yeah.
Okay. Thank you very much.
Thank you for a beautiful talk. You mentioned FGF signaling and Etst1 and cranial neural crest. Has that been linked up the way you linked up Wnt signaling?
No, not at all. It hasn't. There's been, nobody really knows exactly what the links are. So all that data comes from many vertebrates, and FGF signaling has been best studied in frogs. And we don't actually see, in our system, we don't have much evidence, at least in the cranial crest, for FGF signaling, though we do have evidence for BMP signaling, and I'm sure that will come in there. There's probably a little bit of notch signaling that's involved, but perhaps at a later stage. But one of the things I'm excited about with these kind of experiments is we don't really know how these things link up at all. So by finding, by doing this kind of pathway analysis, which is in some ways vacuous, right, because you see all these different components, but in the case of Wnt, we found at least one component that seems to be a link. So maybe some of those other components will be similar. So that's the long-term hope.
I'd like to thank Mariana for this spectacular talk and the audience for the questions. And I'd like to invite all of you to the library for a reception for Mariana, and can continue to ask her questions then.